All right, so we are talking about and finishing up our discussion yesterday. We took our fast plants and we identified, okay, the phenotypes, okay? And just to talk about this backwards a little bit, we had the purple allele and we had the non-purple allele in the cover cotyl. Also, we had the green baby leaves or the yellow. And these are, of course, the phenotypes and the genotypes. We decided to use the leaf color yesterday when we wanted to track the change, okay, of the allele frequency from one generation to the other. I don't think, for me, this is cool. I like doing this with you guys because we could talk about a ditto, or we just did it. We just did this. We crossed the generation. We have new allele frequencies. Everything's in front of us here. Now, I know a couple people yesterday in class that are now home had really great points, and I'm gonna reward you them for saying it yesterday. I don't know why, I was so deep into what I was thinking. Okay, but in any case, we decided to pick this allele. We know that we had basically two alleles: the color of the stem, the hypercotyl, and of course the color of the baby leaf called the cotyledon. So there was two separate alleles. Understand that this lab, I know we're not writing this up yet, but you should have that Punnett square done in your lab book that you're going to put in your lab at some point. We know that Mendelian genetics means you have recessive dominant alleles, and all alleles separate independently of themselves. And whether you get, whether you get the recessive or dominant going to a gamete is 50-50 flip of a coin. Okay, that's law of segregation. Okay, now the main purpose of lab 1 is to figure out if the offspring match the 9:3:3 ratio we would get if we have law of independent assortment and law of segregation, okay? And that'll be done separately with chi-squared. I was using it for a different spin in terms of evolution, okay? And so what we did was we counted the alleles, okay? And this is what we got, all right? We got 23 purple-green, understanding there was There was only 4 phenotypes we should get by the dihybrid cross. And we got 23 purple-green, we got 9 purple-yellow, 8 non-purple-green, and then 4 non-purple-yellow. Okay, eyeballing it, it looks like, man, that could be 9, maybe not, who knows? But what I made yellow here is the fact that we're just going to track the yellow allele because I thought the yellow allele or green leaf was easier to determine than the purple stem, okay? So what I have is, if I'm a yellow leaf, what do I know? If I'm a yellow leaf, I know that I'm a homozygous recessive, okay, for the color of the leaf. So to be yellow leaf, I know that individual definitely has that genotype. That's why the jumping off point for the Hardy-Weinberg calculations to see if there was evolution. And of course, you guys need to know evolution is not just adding new alleles. I know in cladograms you think, hey, I have no jaw, now I have a jaw, I was 2 legs, 4 legs. There's more to it than just adding alleles. It's as simple as changing the frequency is evolution. So in any case, I had 13 people, individuals, not people, but individual plants out of the 44 that was homozygous recessive. And they were Q squared. So 13 divided by 44, 2.95, square root both sides, and I've got my Q to be 0.543, subtract from 1, I got my P. And those are my allele frequencies. Now, we said in the F1 generation that the Q had to be 0.5. Absolutely. Why? Because in our first cross that we did, we crossed, the cross that we did, it was a dihybrid cross. They were all heterozygotes. And so we should expect to be 0.5. Now this is an important point. Some of you guys said this. Maybe it was Charlie too. I'm sorry if I did not. There is a change here from the F1 to the F2. In fact, we say the F2 is a little higher. There is evolution here. The allele frequencies have changed. That doesn't mean they're closer to speciation. That doesn't mean that they have a brand new allele. All it means now is that there's a greater opportunity in the next generation to have what? More recessive alleles. Because now it was 50-50 whether the gametes, pollen, and the eggs had that allele. Now it's greater than 50-50. So there's a greater chance in the next generation because it's greater than 50% that you're going to have even more. And that's where you can get that runaway genetic drift. And so Charlie yesterday correctly said, before I can finish my statement, hey, that's a genetic drift. He's absolutely right. So good job there, Charlie. So this is an example of genetic drift. And why drift? We don't have a lot of individuals here. Small population. Even if we didn't have, the amount of seeds that we have isn't in the thousands. Okay, so, and this isn't huge, is it? But it's just slight enough, which means the next generation, you can see why you would tick up a little bit. And that's why it could lead to 100% recessive allele and possibly get rid of the P, which is the dominant. But also someone else said, and I forgot, and maybe it was Holly or somebody else yesterday, okay, and I'm sorry, I did make a mistake when I was giving these out, and not for the team that's here today, the red team. The blue team, I believe that I opened up the wrong seed packet, and I think I may have got them some other seeds in there. Because of that, and they knew that because they were growing yellow seeds right away. Remember that these guys, the F1 generation, should have been green leaves, purple stems. But I had a test group that I wanted to use, I didn't wind up using, but some people measured those. So believe it or not, not only do we have genetic drift, definitely, we also have some gene flow, okay, happening here. Remember, this can only change for three reasons: gene flow, genetic drift, or natural selection. There's no selective pressure that we put these under to survive better, okay? And we know that because in lab 2, did anyone find a significant difference between the LEDs and the fluorescents? Was there error bars overlapping? Yeah. So we know that that was not a selective pressure, correct? See how cool when you put all those together? So this is due to drift and probably some gene flow, as some of those seeds pollen may have mixed, okay? So good job at home saying that, and I'm sorry I didn't give you the reward that you'd necessary. So there was definitely evolution, okay? And we know these things. Now, we only had 2 generations. Now, of course, I showed you this yesterday, that there's a possibility that it can go either way in small populations. I like that when it keeps going up. So we can either lose that, okay, allele, or go to 100% and lose the other when you have a small generation. That is the drift. Now, I don't think we need that many generations, but it just shows you the idea that small populations, okay, can fluctuate their frequencies just based upon the chance of flipping, 4 alleles in a row, okay? Don't confuse effect. Founder effect and, of course, the bottleneck are not examples of genetic drift. They are conditions that can, what? Lead to. It's a big difference, all right? So that's important. But the key here, party people, is that when we change what? Frequencies, whether it is gene flow or genetic drift or even natural selection, okay, this could lead to genetic diversity decreasing. Now, especially really fast for genetic drift. So we think of evolution as this nice stepwise growing from simple organisms to more complex, but now, of course, I want you to think that in the diversification of life, sometimes we're lowering the diversity. And again, we lower the diversity. The biggest deal here is that, the great example here, if I lose this allele, okay, and that allele was important for some type of reproductive behavior or observation and or mechanical part, then you lose the ability to now mate with someone that used to be part of your population, and now you are a new species by losing alleles, okay? And it could be as simple as changing the color of a feather. Over hundreds of thousands of years, a bird who was a female used to pick out the mate with the brightest red plume. Okay? All of a sudden, you lose that recessive allele that made that red. No longer does that female mate with that male anymore. And even though they physically can mate, she doesn't pick them because it doesn't have the red color, then that becomes a new species. So understand reproductive isolation isn't just a physical barrier or geographic. It can be just changes in your behavioral patterns. Bird call. You can have a recessive allele. Whatever genes are responsible for you making a sound or pitch, and all of a sudden you lose that allele, you can't make the sound the same way. No longer is there a correct mating call, and you've got reproductive isolation. So this genetic decrease that you can get by gene flow, and especially, okay, the randomness that happens in small populations. And understand, in mass extinctions, you're going to start with very low numbers that grow out. So you're going to have tremendous amount of this stuff happening early on. And that's what we call punctuated equilibrium. What's that mean? Okay, so let's make sure we understand these definitions. So we know that we go from what? We know that we have, I'll mark it here somewhere. We know that we have simple organisms lead to more complex. Okay, that of course, natural selection, and a lot of what on the way? Mutations. Mutations over time give us what? New alleles. So we're going to add new alleles in the process. And then we're going to select for them when what? New conditions, we'll call it. You have to have new conditions that select for them. Okay? So that's where we get the what? The cladogram that we've been doing, where we add new, hey, go from here to here and you add new things. So that's important. And I'll do natural selection as NS. Okay? Now, in this process of becoming complex though, all right, we know that there's also some other factors at play here. We have genetic drift, we have gene flow, and continued natural selection, especially if the natural selection is what? What is this very thin line? Stability. Stabilizing selection, right? Stabilizing. Now, why would stabilizing, why is it really important to understand stabilizing? Well, you're going to limit the population's other alleles because you're so selected for a certain number that you lose that diversity. Okay? Maybe that diversity right here, okay, could maintain your what? Reproductive success in changing times. But because you don't have those alleles present, okay, all right, then you could become speciated. So complex we lead to speciation. Now, speciation is, of course, we know that there's a what here that you're crossing over? A reproductive barrier. And of course, there's two types. There's a pre-zygotic, meaning before you can even exchange gametes and sexual, there's, hey, you don't even get close enough because hey, you might do what? Mate to different time periods, temporal. You might mate in different geographic regions, right? Your parts might not fit. Remember the two snails? They have reproductive parts, but because of a change of an allele, they're on opposite sides, the shells, so they can never exchange gametes. Okay, and of course there's post-zygotic, meaning, okay, they do fertilize, but maybe the new hybrid doesn't live long enough to reproduce. Hybrid, what? Sterility, okay? Or they're not, after many generations, they start dying out. They're not viable, okay? But now speciation is different species. Now, we get complex, but then we speciate. Now, speciation is a problem because during this time period, we are lowering the diversity a little bit. So we speciate, become many species. And as I said before, many species have a what? Ability to become extinct as you lose alleles by stabilizing selection, or by genetic drift, or by gene flow. These are the three what? Mechanisms of evolution. That doesn't mean that we don't still keep adding alleles, getting more complex, and there's new what? Always new selective pressures. Always new selective pressures on the horizon that could occur. Now, what about all of this speciation leaves us very susceptible to extinction. Now, of course, there are mechanisms that help maintain the diversity. What are they? What's a major one? Instead of just losing allele left and right, how do we maintain some? What helps maintain some? Heterozygote advantage. Heterozygote advantage. What is that? Because we know here we're lowering diversity. The reason that I can no longer reproduce with a population, with an individual that I used to be able to, is that I didn't add a new allele that I can't read. I lost an allele. The reason why speciation occurs is because you're losing reproductive alleles. But if you're losing alleles, I'm only showing you one. There are many alleles in their genome that are probably being lost and gained in speciation or in genetic drift. So it's important you realize that, okay? But if one is in the reproductive area, we speciate, okay? So diversity is dropping, but heterozygote advantage increases the diversity or maintains. So it's a balance. But if there's not a heterozygote advantage for that allele, okay, it'll be lost. Now I'm going to move this up here. This line was extinction. Now in extinction, we have 5 mass extinctions. You don't have to know exactly the name, when and where. Just be aware why the extinction occurred as it did. It did because the organisms didn't have an allele that best suited that new environment, did they? If you became extinct, it's because you didn't have an allele already in waiting for that new condition. And if we're going to speciate and become many species because we lose some diversity, we try to maintain some with heterozygote advantage, okay? But we become susceptible to this mass extinction. Is there any mass extinction when all animals die? No! Not even the great oxygen event. Some get through, okay? But most die because they're most very susceptible as you lose alleles in diversity and not have that potential black wing, okay, for the peppered moth, they'd be extinct too, okay? So that's important, but this is important. The ones that get through. So we have this big arrow of species, all the species right here, okay? And then we have this tiny arrow of who sneaks through. We snuck through. The big mammals of the Permian all died off. The small mammals, and he even said it in the video, there was one bird-like creature that got through the Permian extinction. It was as big as his pen tip. That's who we were related to. It was a pre-mammal. Why did it survive? It was small. Why the big mammals died? They needed a lot of oxygen, a lot of food to support a warm-blooded individual. This little tiny bird-like kind of running around little creature, okay, didn't need as much energy as oxygen to survive, so it got through. And we had a what? We had an allele. We think we had an allele to survive that hydrogen sulfide event, if you believe in the Kump hypothesis, okay? And we snuck through. Mammals snuck through. Now, of course, the conditions, yes?
What was the Kump hypothesis again?
The Kump hypothesis is that every mass extinction is due to a microbial event where we produce hydrogen sulfide that's so toxic it kills the earth. The idea is that if CO2 goes up, that's what he's talking about. Anytime CO2 goes up, all right, and we know he talked about the biomarkers, okay, that you can find. So there are 5 major places in time periods that we can see in our geological layers and even in ice cores where there are biomarkers where hydrogen sulfide was in tremendous amounts. We have the markers and the evidence that showed HS. We think by producing a lot of CO2, whether that's basalts, volcanoes, or a slamming of a crater, the CO2 levels rose. That CO2 goes into our oceans. CO2 is acidic. The acidity killed off our plankton, our phytoplankton. They no longer can make the oxygen, okay? Okay, and all of a sudden you don't have the oxygen to support the big what? The big mammals. You don't have the oxygen to support the fish in the sea, and there's an incredible die-off. Except for the ones that had the what? That had the alleles to survive those mass extinctions. Now, this is important. You have all these resources out here because all animals died if they didn't have that what? Possible allele to survive the hydrogen sulfide or to survive low oxygen conditions, right? A big 1,000-pound mammal with low oxygen levels. And why low oxygen? The phytoplankton have died. The CO2 went into the water and acidified and killed them. Okay? That's why our coral reefs are declining for the same reasons. But they get through. Okay? And because no one is now, after the mass extinction, the ones that get through and nobody's eating this, no one's eating that, there's tremendous amount of resources. And this is where we have this punctuated equilibrium. This means the speed in which speciation occurred is tremendous. Why? Number one. Yes, go ahead.
Because of all the resources?
Well, there's tremendous resources. That's the selective pressure. The selective pressure is there's plenty of places to get food. Just like when the birds traveled the Galapagos Islands, every island had different, there was food for cactus, there was big nuts, small nuts, tremendous resources for the birds, tremendous selective pressure, okay? But punctuated equilibrium means there was a tremendous amount of speed at which speciation occurs. We know this because the fossil evidence shows a mass die-off. There was a black layer, there was a chalky layer after one of the mass extinctions. That's where all the phytoplankton. There was a black layer, no more phytoplankton, and then of course it regained itself. But we also see in water beds the different types of morphological structures that have started appearing really quickly, which means in the layers, they're very close to each other. We saw new things appearing real quickly, which means this phylogenetic tree wasn't like this. It was more like so many of these things popped up so fast. Why? Number one, a lot of resources, tremendous selective pressure. But two, the amount of individuals who started here are what? Small. Genetic drift all over the place. So there was so much what? If you were a little population together, okay, that got through, there might be another population of the same what? That got through over here. But because you're a small population, geographically isolated, and because of genetic drift, you quickly what? Changed, okay, and lost alleles, and of course had reproductive barriers and became, different parts of the animal. So punctuated equilibrium is this huge speed in which a speciation occurred, which sets up the stage for what? More allele loss, okay, for the next, geographical event. Most organisms don't survive a mass extinction. Most organisms do not survive. Eventually they find their peril because of that, unless there's, some things here. But let's talk about something. So I hope you get that big picture here, okay? So, forces that make speciation possible, the forces that make species susceptible to what? To extinction, okay? Lowering genetic diversity through stabilizing selections or genetic flow and genetic drift essentially decreases a variety of alleles that could potentially give populations the ability to survive. This is why most species become extinct, and this is the reason why older species still prevail today. Think about this. This is why most species become extinct, and this is the reason why older species still prevail today. Think about this. Okay, this is really cool. Command minus. I don't know if you read this or took this in. Maybe you didn't because you're busy with other things. For example, there are 3 elephant species that inhabits Africa, Asia, and they have a distant relative in the manatee. I don't know if you ever looked at a manatee, okay? But they have a face of an elephant, okay? It's kind of cool when you look at a manatee. Now let's look at this phylogenetic tree. It shows time period where speciation, evolution, genetic drift, gene flow, but definitely speciation occurring here. Now, check this out for a second. These are the 3 living, okay, elephant species. Okay, all of these guys have gone extinct. Notice the line here is the current timeline. These guys haven't made it. This is where we last found that fossil. Okay, so what we're seeing here is what? The manatee is still here. Still survived. Elephants, and so many of them did what? Went extinct. Right here is punctuated equilibrium. You see this punctuated equilibrium? Small time periods, we have so many species, which means we had tremendous amount of speciation, genetic what? Diversity dropping a little bit to cause that. And what made them speciate, made them susceptible for what? Extinction, except for the last 3. These guys didn't speciate. They're still very successful. Why are these guys so successful? They maintain their genetic diversity while these groups didn't. It's pretty cool to look at that. Okay, well, we're still good, right? No, I still have a few more minutes left. Okay, so in any case, let's look at the humans. Okay, and interesting enough, monkeys were kind of 4 legs, so to speak, and all of a sudden there was an adaptation or a change where we started walking. And some people say that the walking was a bigger adaptation in the the humans, or hominids, sorry, than the big brain later. And interesting enough, okay, adaptations for walking bipedal occurred a lot. That was more important than the big brain. So we became human-like when we became bipedal. And we don't know exactly, but anthropologists look at when they saw these fossils of our hip bones changing. Okay, of course that's a mutation, and of course there was a selective pressure. What was that selective pressure? Okay, so yes, we have the what? Remember we talked about this, the skull size, a punctuated equilibrium. This skull changing occurred very quickly in our last part of our life, as or at least in our geographic or lineage of humans, hominids. This occurred really quickly, okay? And look how many perished. We survived. Why? Speciation, genetic drift, gene flow. To be a different species, meaning you had to limit your what? Your alleles, make you more susceptible to the extinction, okay? But what's really important is that the bipedalism was an adaptation that occurred long before our large brains. What possible selective pressures made bipedalism an advantage? Or do you think that bipedalism was just a non-adaptive form of evolution? Was that a gene flow thing? Was that a loss of an allele? Well, we think it was natural selection, okay? And there's a couple different major theories, and it's very interesting why we became up 2 feet. Well, number one, the amount of trees that we lived in decreased, okay? Supposedly the landscape changed as the global warming occurred during our time period. And so we needed to walk far distances for food. So walking on 2 legs is more efficient than 4 legs. We save more energy, which you say, well, that's not a big deal. How much more do you save? But if it's a 2% or 3% advantage over 1,000,000 years, the people that save more energy don't starve to death as much, okay? Also, we noticed that we don't have curved fingers. We talked about that in another one, right? Because we're not climbing anymore. Why would we go to bipedalism? Well, people think that instead of climbing trees and getting our food that way, we needed to carry our food back to our people. How do you carry food back if you're working on all 4s? It stays in your mouth. This is a more efficient way to gather food. Therefore, you what? Have a better way to survive. You increase your relative fitness that way. All right. I didn't go over the forms, but I definitely wanted to link some of the things we talked about. All right. And so, really important you guys understand that major oxygen event. Also, real quick before we leave, okay, there is this, please look at this before you leave. I want to put this on the big screen. There is eukaryotes for prokaryotes. So we're starting out big, and now we're going small. Eukaryotes, are they an evolutionary, which one's older, the prokaryotes or the eukaryotes?
Prokaryotes.
Prokaryotes are older, We're going to talk more about these guys. But there's lineage now on simple cells. Now think about efficiency, okay? Endosymbiotic events, what are those? What's an endosymbiotic event? Well, what was the lineage? And I'm going to draw this on here because I know we have to go, but if you look at a phylogenetic tree of the earlier organisms, okay, this is now going way back in time. We have bacteria, we have archaea, and then we have eukaryotes. I know you can't see that brightness here. So this is bacteria, archaea, eukaryote. Okay, and this is bacteria. Now these guys are considered to be prokaryotes. The archaea are like bacteria that lived under really, really stressful conditions. They've changed a little bit. They can live in hydrovents. They can live on low temperatures. This, of course, is bacteria. Okay, and one of these is cyanobacteria. Remember that last question? Yes. Cyanobacteria had that ability to photosynthesize. Grab CO2, grab what? The light, grab water, and make what? Sugar-based compounds, and produce oxygen. And it was that oxygen production that almost killed all things. But it's that oxygen that we're going to later learn about that gave us the ability to make more energy, be more energy efficient. When did eukaryotes start? Here's where we think. It's when somehow a prokaryote, which we know is smaller, inefficient, engulfed a smaller organism and then used it symbiotically to make energy for itself. This event is when we engulfed and became mitochondria, or we engulfed the cyanobacteria, okay, plants. And we know this to be true because these little organelles, they have their own DNA. We know that prokaryotes have circular plasmids. We're a eukaryote. We have a what? Membrane-bound nucleus, and where we can see the what? Chromosomes. But when we look at a mitochondria, when we look inside it, we actually find plasmids. It was once its own what? Organism. Just like in a plant cell, you'll find a chloroplast that has its own plasmids. So we became bigger, more complex cells, we think at this junction, when we engulfed the chloroplast, or we engulfed what now is a mitochondria, that gave us the energy efficiency to be more complex. That's the connections I wanted to make. Have a great day. See ya.